Okay, so as I was, we're going to try to address these three different questions. How different are the genomes among species? Is a larger genome more complex? And if there is a correlation between genome size and organism complexity. And we were saying that we are going to review some examples of primitive organisms, as well as organisms that exist today. And so in this picture, we have a diversity of organisms going from bacteria to mammals. And then we were asking whether the genomes of those animals and plants or organisms are similar or not. How different are they? And we have, for example, bacteria that are unicellular organisms and fungi that can be unicellular as well or multicellular. So how different the genomes are. And we were looking at this picture here where we have bacteria that have very small, the smallest genome described, but they can also have a little bit larger genome. So there's a wide range of sizes here. Then we have protists, which are unicellular organisms, only one cell, but the genome of these organisms can be very large in millions of base pairs. So they can have a very large, a lot of DNA in their nuclei. And then we have fungi, and we have here some examples of plants, insects. We have fish, amphibian, reptiles, birds, and mammals. And as you can see, if we go down to mammals, which are very diverse species, the range of the genome is not that wide. I mean, we don't have very small genomes and we don't have very large genomes. Most of the genomes of mammals are around this size here. And there are some frogs that may have a larger genome than humans, and even some plants that can have much larger genome than humans. So that means probably that the size of the genome doesn't necessarily correspond or correlates with the complexity of the organism. So here, asking to this question, how different the genomes are, we can see that the size of the genome is very variable, and it doesn't really correlate with the size of the organism. So we have small organisms with very large genomes, and we have very large organisms with relatively small genomes, okay? So just imagine we have a mouse, a human, and a whale, and we are very different in size and complexity, but our genome is very similar in size, okay? So let's take this example. We have, on one hand, we have first the yeast, which is called Saccharomyces cerevisiae, which is the yeast that is used to produce beer. So this is an eukaryotic cell. They are unicellular. They have 20 million base pair genome, and they have 6,000 protein coding genes distributed in 16 chromosomes. Okay, so this is the smallest eukaryotic cell. Now let's have a look to this worm. It's a 1.2 millimeter worm. It's a very tiny worm. It's a nematode. It's called C. elegans. It's a multicellular organism. It has different cell types. It has intestine, eggs, gonads. So it's a very complex organism compared to the yeast, because yeast is only unicellular. And it has approximately 1,000 somatic cells and 1,000 germ cells. So let's say that this little worm has 2,000 cells in total. And DNA is now 100 million base pairs. So we go from 12 million to 100 million base pairs. It's more complex. Its DNA is much larger, and it has a lot more protein coding genes, 20,000 compared to 6,000. And it has about 12 chromosomes similar to this. So, but what happens when we go to Homo sapiens, or the human being? So I'm drinking the beer that the yeast produced. So we have a lot more cells than these organisms, the nematodes. Our genome is much larger. So C. elegans has 100 million base pairs, And in humans, we have 6.4 billion base pairs. So it's a lot larger. But the interesting thing is that C. elegans has 20,000 protein coding genes. And humans, we have 30,000 protein coding genes. So if we look at the difference in the genome size, we go from 100 million to 6.4 billion. That's a huge difference. But when we look at the protein coding genes, there's not such a big difference. There is a difference, but not that much. It doesn't correspond to the size of the genome, okay? And we have other examples, like in this case, we have some plants, some trees. First, we have the influenza virus. It has 13,500 base pairs. We have walnut tree, 650 million base pairs, human 3 billion, and we have these two big, very large trees that have 8 billion, and this one that has 27 billion base pairs. And here we can think, okay, the genome size probably correlates with the size of the organism, because these trees are maybe very tall, very high. But what about this guy? So this you probably know, this is ajolote, the axolotl. It's a salamander. It's that big, it's very small. We can take it in our hands. And its genome, it's 28.4 billion base pair. It's much larger than the human genome, but it's a smaller organism. Is it more complex or not? We don't know. I mean, it's more complex in some things, but it's a small organism. So the size of the genome, it doesn't correlate with the size of the organism. Okay, that's the first point. There are some work that has been done using mathematical models to estimate the size of dinosaurs, for example. And this one, the Triceratops, correspond to this family here, and T-Rex correspond to this family here. Yeah, this one. So basically, here, what you can see is that, okay, estas se las explico en español, igual que en la mañana, para que me entiendan un poco mejor. Pues aquí lo que hicieron fue, usando modelos matemáticos, calcularon la probabilidad de qué tamaño serían los genomas de Triceratops o de diferentes dinosaurios de esas familias, ¿no? Y vieron que, por ejemplo, el Triceratops, lo que representan estas barras, el grosor de la barra, no el largo, sino el grosor, representa genomas más grandes. Entonces, este es un genoma más grande que este. Estos son los genomas de los animales como el Triceratops, y estos son los genomas como, por ejemplo, los T-Rex. Y entonces aquí lo que ellos sugieren es que en los organismos, en los dinosaurios, los genomas, el tamaño de los genomas no correspondía necesariamente al tamaño del organismo, ¿vale? Entonces también ya hay evidencias no concretas y concluyentes, pero que sugieren que podría ser lo mismo que vemos hoy en día en los organismos de nuestra época, ¿no? Hay otro ejemplo, que también se los explico rápido en español, de un pececito que se llama lungfish, que este estudio lo hicieron en fósiles de ese animal que tenían 400 millones de años, 300, 200, 100 y hasta la actualidad. Y lo que vieron es que los organismos más antiguos, o más bien hace más millones de años, había más variabilidad en las especies. De ese mismo animalito había como 17, 18 géneros y más especies, ¿no? 18 géneros. Conforme nos acercamos hacia acá en el tiempo, o sea, hace 300 millones de años, ya solo había como 5 géneros diferentes. Si llegamos a 100 millones de años, ya solamente había 2 géneros. Y cercano a nuestros días también había 1 o 2 géneros diferentes. Y lo interesante es que eso se correlacionó inversamente con el tamaño de las células del animal. Es decir, los más primitivos, basados en los fósiles, tenían células muy pequeñas. Y los más nuevos, en la evolución, tenían células más grandes. ¿Y cuál es la relevancia de eso? Que hay datos muy consistentes actuales en los que se ha visto que el tamaño de las células de un organismo correlaciona con el tamaño de su genoma. Organismos con células chiquitas tienen genomas chiquitos, con células grandes tienen genomas grandes, y eso parece ser el caso desde el punto de vista evolutivo, les digo, con este análisis de tamaño de las células y variabilidad de géneros en esta especie de peces, ¿no? Otro ejemplo se hizo con diferentes géneros de insectos. Entonces aquí lo que podemos ver es que de la línea del 0 para abajo representa la variabilidad de cada género. Por ejemplo, aquí tenemos dípteros, y dípteros hay un montón. Aquí, 250 millones de años atrás, había poquitos, y fueron evolucionando hasta que hoy en día tenemos una gran diversidad de dípteros. Y lo que vemos de la línea para arriba es el tamaño del genoma. Entonces, realmente son genomas que no pasan de un tamaño, de aquí son dos, no me acuerdo cómo lo clasifican aquí, pero son tamaños relativamente pequeños, ¿no? Si nos vamos hacia la derecha, en estos géneros que están aquí, donde vemos que desde las especies primitivas hasta hoy no ha habido gran diversificación, o sea, no hay tantos géneros y especies, y lo que vemos es que algunos de ellos tienen genomas más largos, ¿no? Más grandes, como estos de aquí, por ejemplo. Están por arriba de la media de los genomas de especies muy diversas. Entonces, lo que se sugiere es que también cuando una especie tiene muchos géneros, hay muchos géneros diferentes y muchas especies, los genomas son chiquitos. Y cuando solamente hay poca variabilidad en género y especie, los genomas son más grandes, ¿no? Ese es también nada más un dato evolutivo. Ahora, veamos este ejemplo ya de organismos más recientes, ¿no? Que conocemos hoy en día. Entonces, por un lado, primero quiero que se enfoquen en la... déjenme ver si puedo... Quiero que se enfoquen en la gráfica del lado izquierdo. So we are going to concentrate in bacterial genome. So in this table, we have a list of different organisms, and we have the genome size in base pairs, and we have the number of protein coding genes. So here, what we can see in bacteria is that small bacteria with small genomes have only few protein coding genes. So these bacteria, that is the one with the smallest genome, 160 kill base pairs, it has only 182 base pairs. What happens if we go further down? We see how the genome size increases, 160, 580, 1.7 million, 2.7 million, 2.9 million, 4.3, okay, and 13. So the number of base pairs, the amount of DNA present is increasing as it increases the number of protein coding genes. So that's very nice. There is more DNA, there are more protein coding genes. But what happens if we look at eukaryotic cells? So we have two examples first, the top one and the bottom one. This is a fish, that is this one here, Fuju rubripus, and we have the chimpanzee down here. So if we look at the genome size, this one, the fish, has only 400 million base pairs, whereas the chimpanzee has 3.3 billion. So it has a lot more, it's giga base pairs, and here it's mega base pairs. So the chimp has a lot more DNA than the fish. But when we look at protein coding genes, they have about the same. You see, the fish has 19,000 protein coding genes, and the chimp has also 19,000. So that's strange. So how is it possible that two different organisms that are so different in complexity, in size, in many things, they have similar protein coding genes? And then if we look at this tree here that is called Populus trichocarpa, so it's down here. So it has 500 million base pairs, similar to the fish, but it has 46,000 protein coding genes. A lot more protein coding genes than the fish that has similar genome, and a lot more than chimpanzee. And if you see here, there's also the corn, maize. We have a large genome and we have a large number of protein coding genes, but some others, the other animals, there is no correlation between the genome size and the number of protein coding genes, okay? Okay, so if we go back to this comparison when we see prokaryotic cells, eukaryotic cells, the genome is very small here, as you can see here, and it's much larger and complex in eukaryotic cells. And then what determines the size of the genome in bacteria and in eukaryotic cells? So here, what I wanted to say is that in bacteria, most of the genome is composed of protein coding genes. It means the vast majority of the DNA in the bacterial chromosome will have information for coding proteins. But in bacteria that are pathogens or obligate symbionts, they don't have what we call pseudogenes or repetitive elements. But bacteria that live free, they have more complex and larger genomes. Okay, so bacteria that live inside another organism, they have small genomes and not very complex. And bacteria that are free living, they have larger genomes. Okay, so but anyway, in any of these cases, the size of the genome depends basically on the number of protein coding genes. But what happens in eukaryotic cells? So the first point is that eukaryotic DNA contains centromeric and telomeric sequences. And what are these sequences? So if you remember, the genome of in bacteria, the chromosomes are circular, and in eukaryotic cells, they are linear. So one of the big differences is that there are two structures in eukaryotic cells that are not present in bacteria. And these are called centromere here and telomeres there. So what are these structures? So centromeres, if you remember, when the cell is going to divide, it makes a copy of the DNA. So each chromosome is going to be copied. We have two copies of the DNA, and they will stay together. The region of the DNA that keeps together the two chromatids, it's called the centromere. And on top of that, the centromere is going to be very important because the microtubules from the mitotic spindle will trap the chromosome, pull them apart. Okay, so that's a very important structure. It's not present in bacteria. And the other one is the telomere or telomeric sequences, which are at the end of the telomeres. So what is the relevance of these telomeric sequences is that at the ends of the chromosomes, just give me a second, I'm going to share you. Okay, so if we have a circular chromosome in a bacteria, it's a double-stranded DNA, but in eukaryotic cells, we have linear chromosomes. So the first thing we can notice is that there are these ends here that are free, and they are not present here. So it's like these regions have to be protected. Se los voy a poner un ejemplo en español. Imagínense que es una agujeta y en los extremos de las agujetas tienen esos plastiquitos que les ponen, porque si los quitamos la agujeta se enrolla o se rompe. Entonces el DNA, al ser lineal, tiene dos extremos que quedan desprotegidos. Y entonces en esas estructuras terminales de los cromosomas se llaman, hay unas regiones que se llaman telómeros, que luego lo vamos a ver con detalle en una clase, pero esas regiones no están en un genoma circular porque no tiene extremos, ¿de acuerdo? Entonces, regresemos a... voy a adelantarle las datos donde íbamos. Okay, so these two structures are very important for eukaryotic chromosomes. This one, as I told you, just to keep the chromatids together and then to pull apart the two chromatids so that they can be segregated to the new cells. And then at the telomeric sequences, it helps to form this T-loop structure. So basically the DNA will form a complex with many proteins here that will protect the end of the chromosome. And we are going to see that examples of that in another class later. But just remember, there are two structures that are different from prokaryotic cells, from eukaryotic cells. But all the eukaryotic cells have centromeric and telomeric sequences. So these sequences do not contribute to the difference in the genome in eukaryotic cells. So there should be something else that makes the difference among eukaryotic cells. And basically, we have some sequences that we call repetitive sequences. So we have two classes of repetitive sequences. One that are small, they are called short tandem repeats. So we can have AGAT, that's a very short sequence for nucleotides, but that sequence can be repeated 7 times in a row, or 6 times, or 4, or 19, or 14. And different individuals, we have different numbers of repeats. And that's something that appears in all the genome of eukaryotic cells. And then we have other sequences that are called long or short interspersed elements. So those are sequences that are not small like this, but they are much larger, and they can be inserted in different parts of the genome. So to have a more clear idea of that, let's imagine that this is a chromosome here. So in a chromosome, eukaryotic chromosome, we will have protein coding genes here and here. We will have the telomeric repeats. So at the end, to both ends of the chromosome, we have these telomeric regions. We have the centromeric regions here, and we have these STRs that are these small sequences that I was telling you, STR, short tandem repeats. So we have some of them here, we can have more there, and we have these long or short interspersed. So these ones here on the right are like line and sin. This is the abbreviation of the code. So we can have all those sequences in a genome, and they are not coding for proteins, okay? But what happens in all the other parts of the genome? So there are other parts of the genome where there are no protein coding genes, there are no telomeric regions, there are no centromeric regions, there are no long interspersed repeats, there are no short tandem repeats. So what is there? And now we know that eukaryotic genome contains a large amount of functional non-coding RNAs. So there are regions in the genome that are converted into RNAs, but those RNAs are not converted or are not used to produce proteins, but they stay as RNAs, and they can be classified as small or long non-coding RNAs. And they play a very important role in regulating the function of the protein coding gene. So these are called regulatory RNAs. And something interesting is that some of those RNAs that are called long non-coding RNAs, that first it was thought that they were just they had no function. Now we know that those RNAs that are very long, they can form very complex structures, and they can have a regulatory function to regulate the function of protein coding genes. But also, now we know that those non-coding RNAs can actually go to the ribosome and be used by the cell to produce small peptides. What's the function of those peptides? We still don't know clearly, but there are some peptides that are produced from these non-coding RNA. And this is just an example of all the different types of non-coding RNAs. So we have the small interfering RNAs, microRNAs. The ones that we were talking about are the long non-coding RNAs. And there are many, many different types of RNAs that have different functions in the cell. And we also mentioned that some of those long non-coding RNAs can produce small peptides that are called micropeptides. And some of them have functions that are already been characterized. So the picture is getting more and more complex because there are some regions in the genome that we thought were useless. Now we know that they can have a function. So to finish with this part, so the 3 questions: how different are the genomes among species? As you could see in some of the slides that I show you, the genomes are very different in size. And if we compare prokaryotic cells with eukaryotic cells, also there are more complex genomes because they have a lot of non-coding sequences. Then is a larger genome more complex? Not necessarily, but most of the large genomes are complex because they have a lot of non-coding regions that can play roles in the regulation of the coding regions. And finally, is there a correlation between the genome size and the complexity of our organisms? First, we can say that no, because as we can see, as you could see in one of the slides, there are very small organisms that are not very complex, and they have very large genomes. But I will say that the complexity of an organism is determined by the three aspects that we're seeing here. The total DNA content, so the number of base pairs, but also the number of protein coding genes, but also the amount of non-coding DNA. So I think these three elements together are the ones that contribute to the complexity of the organism. So there may be two organisms that have similar genome size. They have maybe similar number of protein coding genes, but they have very different amount and type of non-coding DNA, and that will make the difference. Even though the genomes are similar in size, they are similar in protein coding genes. If they have a lot of differences in non-coding DNA, that will make a big difference in the complexity of the organism. Okay, so bueno, con esta parte termino lo que tiene que ver la parte que les iba a platicar en inglés. Ahora lo que vamos a hacer es vamos a platicar un poco en español de si es que ustedes leyeron algo y de lo que pudieron cacharle ahorita con las diapos, y si no, ahorita se los voy a ir explicando. Entonces, ¿el tamaño importa o no importa? Desde el punto de vista del genoma,